triple-negative breast cancers, or TNBC, is one of the most aggressive breast cancer forms, accounting for significant portions of breast cancer mortality. Capsaicin is a bioactive component found in chili peppers. Studies have shown capsaicin can induce apoptosis in cancer cells by influencing key molecular pathways, including the upregulations of BAX, a pro-apoptotic protein, and the downregulations of fatty acid synthase, or FASN, an enzyme often overexpressed in TNBC. This study aimed to extract and analyze capsaicin from red peppers and determine its effectiveness in influencing cell proliferations, apoptosis, and gene expressions in the TNBC cell line, MDA-MB231. It is hypothesized that capsaicin treatment would promote apoptosis, upregulating BAX expressions and downregulating FASN expressions in breast cancer cells. Capsaicin was extracted from dried red peppers using supercritical fluid extractions. I perform another traditional extraction method by soaking red peppers in ethyl acetate, where the extraction was then roto-evaporated and resuspended in methanol. We call this EAM extraction. From a PLC analysis, all extraction confirmed the presence of capsaicin. EAM extraction achieved the highest concentrations, which can be attributed to the solvent properties of ethyl acetate for extracting nonpolar compounds like capsaicin. The SFE extraction shown to be more selective, however, had a much lower concentrations and recovery rates, which may be linked to technical challenges with maintaining the necessary pressures. All the pressures applied during SFE were insufficient to achieve the best extraction yield. Unfortunately, all extraction did not meet the required concentrations for cell treatments. Therefore, capsaicin standard were used in the rest of the project. Moving to our cell experiments, I treated MDA-MB231 cells with capsaicin at various concentrations and assessed the cell viabilities over 24 and 48 hours. The MTT results reveal significant cytotoxic effects at concentrations of 170 and 200 micrograms per ml after 24 hours treatment, and at 100 micrograms per ml and above after 48 hours treatment. This indicates a time and dose-dependent response within breast cancer cells, with effective dosage needing to be within a 48 hours time frame, and the dosage needed to be at 100 micrograms per ml and above to achieve significant cytotoxic effects. I then determined the cell states and cell apoptosis by using ethidium bromide and acridine orange staining, where the green cells were the live cells, the yellow cells were apoptotic cells, and the red cells were necrotic cells. After 48 hours treatment, the EVAL assay showed a significant increase in apoptotic cells at 100 micrograms per ml compared to the vehicle control, with even more pronounced effects at 200 micrograms per ml. This supports capsaicin's cytotoxic impact in a time and dose-dependent manners, with higher concentrations will result in a more pronounced apoptosis. I further measured the capsaicin effect on gene expressions by measuring the two BAX and FASN gene using RT-qPCR. Different from the expectations, capsaicin did not significantly alter the expression of BAX and FAS gene, especially the expression of BAX were decreased. This suggests that alternative pathways may be at play in mediating capsaicin-induced apoptosis in TNBC cells, highlighting the complexity of cellular signaling. Future studies could explore additional extraction techniques to optimize capsaicin yield and reduce extraction time, make it more suitable for large-scale applications. Furthermore, investigations of alternative molecular targets and side effects of capsaicin treatments may reveal a new therapeutic treatments in TNBC. Additional research is needed to fully understand the underlying molecular pathways and optimize capsaicin therapeutic potential for breast cancer treatments.